I want to talk about Alzheimer's, the most common neurodegenerative diseases in recent years. The pathological features of this disease include extracellular amyloid beta, catenating, senile plaques, and intracellular hyperphosphorylated tau, catenating neurofibrillary tangles, neuroinflammation, synaptic loss, neuronal death, neurocortical atrophy, and progressive decline in cognitive function. Currently, the FDA has approved only 5 drugs for Alzheimer's treatments: donepezil, rivastigmine, galantamine, tacrine, and memantine. The first 4 drugs are acetylcholinesterase inhibitors, and the last one is an N-methyl aspartate antagonist. Low acetylcholine levels have been found in Alzheimer's brains. Acetylcholine is known to be associated with memory, learning, and cognitive function. Therefore, the deficiency of acetylcholine is linked to Alzheimer's, and these drugs aim to reduce acetylcholine levels. Clinical studies show some other approaches to Alzheimer's diseases, such as acupuncture, behavioral training, and brain stimulation, including deep brain stimulation, repetitive transcranial magnetic stimulation, and transcranial electrical stimulation. However, the current treatments have not achieved sufficient therapeutic results, indicating a need for new treatments. In recent years, stem cell therapy has gained interest as a potential regenerative treatment for neurodegenerative diseases due to its ability to regenerate neural tissues, stabilize the neural networks, provide neurotrophic support, and alleviate neurodegeneration at different neural circuit levels. In a study, experiments were conducted using a type of multipotent stem cell called neurostem procenter cells with the ability to proliferate, self-renew, and generate new neurons and astrocytes in mice and rats. The total analysis of this experiment was the subject of the study. The neural stem cells group was compared with the control group, and it was observed that the neural stem cells group could significantly improve cognitive function. Alzheimer's disease is a progressive neurodegenerative disease, and in this study, cognitive function was the main focus. The pathological features of Alzheimer's diseases include extracellular amyloid beta accumulation, the presence of intracellular hyperphosphorylated tau, neuroinflammation, and synaptic loss. Therefore, changes in these features were compared with the neural stem cells group. Looking at these pathological features, the accumulation of amyloid beta is considering the fundamental pathogenesis of Alzheimer's diseases leading to synaptic loss, neuroinflammation, and cognitive impairments. Meta-analyses have reported that after neural stem cell treatment, amyloid beta accumulation was lower than in the Alzheimer's disease group. Regarding synaptic density, synaptic damage in the neocortex and limbic system leads to memory impairment and is typically observed in the early stages of Alzheimer's disease. Synaptophysin is a specific protein found in the synaptic vesicle membrane and plays a role in the formation of synaptic vesicles and dendritic spines. Synaptophysin was used to assess synaptic density in the study. It was suggested that neural stem cells supports the recovery of synaptic density, weakens synaptotoxic properties, and promotes synaptic plasticity. Neuroinflammation is not typically considered causative on its own but contributes to increasing disease severity. IL-1 beta, which exacerbates the brain's inflammation. The anti-inflammatory effect is believed to be a result of one or more other Alzheimer's disease pathologies or related risk factors. Neuroinflammation is not typically considered causative on its own but contributes to increasing disease severity. IL-1 beta, which exacerbates beta-amyloid and tau pathologies, has been identified as a master regulator within the brain's inflammation region. Impairments in IL-1 beta may delay the onset of neuroinflammation and neurodegeneration. However, in the study, it was observed that there was no significant change in IL-1 beta expression. But when macroglia and astrocyte density were measured, it was seen that they decreased after neural stem cells transplantation, suggesting that neural stem cells might inhibit neuroinflammation. Tau proteins are microtubule neural proteins with a macrotubule binding region involved in the polymerization and stabilization of the microtubule array to maintain the integrity of the cell skeleton. Hyperphosphorylated tau forms NFTs and accumulates in the cytosol, losing its ability to maintain cell structure, leading to cognitive dysfunction. In the study, it was found that the neural stem cells group had lower p-tau levels. In conclusion, this study investigated whether neural stem cells could be used to treat Alzheimer's disease. It confirmed that neural stem cell therapy could improve cognitive function in preclinical models of Alzheimer's diseases. This indicates that the improvement of a neurotrophic factor called brain-derived neurotrophic factor in the brain can improve the condition of Alzheimer's disease. Brain-derived neurotrophic factor is expressed by glutamatergic neurons and glial cells, including astrocytes isolated from the cortex and hippocampus. Brain-derived neurotrophic factor is a neurotrophin that modulates stem cells survival, neurogenesis, neural differentiation, branching and survival of differentiated neurons, and the formation and maturation of dendritic spines and synapses. It is a dimeric polypeptide growth factor. Thus, the improvement of brain-derived trophic factor also affects the enhancement of learning and memory. Additionally, it was observed that the effectiveness of neural stem cell treatment was increasing by using nanoformulations and genetically modified injections. But allogenic and xenogenic therapies were found to improve cognitive function. I hope that this translation maintains the structure and meaning of the original text. Please note that medical terms and concepts have been accurately conveyed while preserving the appropriate terminology and syntax. The heterogeneity in the analysis in the study could not be significantly reduced, and it is possible that human physical conditions might show different effects than mice rats. More research needs to be done for this.